This podcast is brought to you by the Albany Public Library main branch and the generosity of listeners like you. What is a podcast? God, Daddy, these people talk as much as you do. Razib Khan's Unsupervised Learning.
Thanks for listening to the ungated version of the Unsupervised Learning podcast. If you want to read some essays on some of these topics, please check out razib.substack.com. Again, that's razib.substack.com. Thank you.
10% of pediatric cancer is linked to a single gene variation. These variants can be detected in embryos before pregnancy begins. Orkin's whole genome reports can help mitigate your child's risk for cancer by screening for 90-plus genetic variants linked to pediatric cancer. Discuss embryo screening and IVF with a genetics expert. Hey everybody, welcome to this episode of Unsupervised Learning. I am here today with my friend Chad Niederhuth, who is a scientist in the agricultural sector. He's a geneticist, and we're going to be talking about plants and academia and industry. I talked to Chad a couple of years, or no, was it 5 years ago? Is it 5 years since I had you on the Insight?
Pre-pandemic.
Whoa, okay, like we're old, but I just feel like it wasn't that long ago, but like 5 years is not, you know what I'm saying? Say?
Yeah, it's not true.
It was, my kids were a lot younger, and your kids were, well, some of them were not
even born yet.
Okay, so it's a different world. It was pre-pandemic. And we talked about, I think, mostly epigenetics and stuff like that. Maybe we'll touch on that a little bit, but mostly what I want to talk to you about is plant genetics, and also, later on we might talk about industry and academia and some of that stuff. I think a lot of the listeners know that, genetics, Mendelian genetics, has an origin, with plant breeding, Mendel's garden and whatnot. Today, we, pretty much associate genetics, as I think with humans and the medical stuff, and, it's the Human Genome Project. But there's a whole world out there besides humans, and plants have their usage. Just to state the obvious, we eat them directly or indirectly. So it's a pretty big deal. And we've talked about the Green Revolution and GMOs and other things, Chad. Can you talk really quickly about your background, why you went into plant genetics, and give some high-level differences between plant and human?
Yeah, certainly. So my background really, I grew up on a farm. My family still farms. And so that's kind of my introduction to plants and why I really got into them. And then when I was an undergrad, thinking of going home and returning to the farm, I started working in a lab and really got bit by the microbiology genetics bug, and went on to grad school, did PhD working Arabidopsis, a model plant species, kind of the mouse of the plant world, working in the microgenetics of Arabidopsis. Then went on to do a postdoc where I was really switched to full-on genomics, and especially epigenomics, working on the things like DNA methylation and chromatin. And then for a while, for some years, was assistant professor at Michigan State University, where I had my own lab, still working on questions of chromatin and the epigenome and really how that had evolved in plants. And then, for a variety of reasons, especially family reasons, about a year or so ago, left for a position in the agricultural industry sector.
Yeah, so in terms of plants versus humans, a lot of organs are asexual. They're not even diploid. That's not even a question. But, with humans, obviously you have two copies of the genome, so that's how Mendel's laws work. With plants, that's not always true. There are polyploid plants in the agricultural space in particular. Can you talk about how that affects your work and how you anticipate the way genetics is going to work?
Yeah, so depending on what you're working in plants, it's either very similar to human genetics or it can be quite different. So, to give kind of a few basic examples of how different plants can be, probably many of your listeners are familiar with mitochondrial DNA, right? You have this organelle that has its own genome that's separate from the nuclear genome and is passed only down through the mother. Well, in plants, you also have mitochondria, but you also have the plastids, the chloroplast that has its own genome, which was from a separate endosymbiotic event of a ancestral bacterial algae. And so that is also typically passed through the female in plants as well. So you have this an additional source of inheritance besides mitochondria. But then within the nuclear genome, you can have high levels of polyploidy. So where you have, humans have typically 2 copies of each chromosome. Plants, you may have many more, depending on the species. And those extra copies of genome, sometimes they're almost exact copies of themselves derived from the same species. That's called autopolyploidy. There can be cases of hybridization between plant species. So if you have 2 species crossed together, and now you have essentially a new species with carrying sets of chromosomes from both, that's called allopolyploidy. And a lot of our critically important crop species are actually allopolyploids. So, for instance, if you consider something like wheat, that is a hexaploid, that meaning it has 3 sets of it has 6 chromosomes, but 2 of those chromosomes are derived from one parent, 2 from another parent, and 2 from yet another parent. 2 closely related species, but distinct species. And this can be quite complicated because, for instance, let's say you want to do like a mutagenesis or CRISPR. You want to modify a gene, right? That may mean that you are trying to modify not just one locus in that genome, but actually multiple loci that you have to modify in order to achieve the desired result. So that can complicate things on kind of the more molecular genetics end. Certainly it plays a complicating role when you're trying to breed as well. And there can be all kinds of crazy things like crossover between these chromosomes that are derived from different parents, and it can really complicate matters. And it just depends, certainly for the genomics end, which is a lot of what I do. If you think of things like genome assembly, that is much more of a difficult task typically in plants, because you can have, when you're working with these polyploid crops, parsing out which segment, which piece of DNA comes from which chromosome is a real challenge.
Yeah, I do want to say, I act like polyploidy is like super freaky, but there is, I believe, some of the teleost fishes, like the salmonids, are tetraploid. So it can exist in metazoans, it can exist in animals, but in general, it doesn't. And I'm just curious why it's so common in plants, but it's not common in animals. So in animals, we know Down syndrome, trisomy 21. They have 3 copies instead of 2. The dosage gets all messed up, et cetera, et cetera. I'm assuming like you can't just ploid plants willy-nilly, but it seems they're way more tolerant of it. Is there like a hypothesis for why?
I'm not sure if there's a set an overarching singular explanation, but I think one of the big factors, and this has kind of been my speculation, is that plants don't have as as deterministic of development as, say, a lot of your metazoans do, right? I mean, and so they're a lot more tolerant of things being messed up. They can, survive or persist when you alter development quite drastically, and still survive. And so many polyploids, you can create polyploids and sometimes still be sterile. So obviously those are ones that are not going to pass on. So there's definitely blocks to polyploidy that happen within plants. There's kind of a limit to what you can do, but I think a lot of it boils down to not having the development being more plastic than mammalian development.
Yeah, so I guess what you're saying is they don't have as many necessary failure points. If they're more plastic, there's multiple paths, and so it de facto results in some sort of redundancy or anti-fragility or more robustness, right?
Yeah, I mean, to give kind of like a counterexample, or to give kind of a example, there's quite a few plants species that reproduce through apomixis, right? So this is where like you still are producing a seed or still kind of going through initially the pathways of sexual reproduction, but essentially you end up with a seed that's going to be derived entirely from the maternal genome rather than having any input from the paternal side. This exists, I think. There's like some lizards and others that do kind of a similar thing, but many of the cases of apometic plants are actually triploids, where you have 3 copies of each chromosome, and so there's an example of where you've altered the dosage of the genome in a way that is actually directly affecting reproduction, but the plant can still persist through it, taking this apometic route, even though now it's no longer going through normal sexual reproduction. And so, that in humans, right, that something like that would just be lethal, non-viable. So that's kind of what I mean by that plasticity, that they seem to developmentally and reproductively, I think they tolerate a lot more things.
Well, okay, so you were talking about humans and plants. Obviously, plants brackets a lot of lineages, and this isn't in the notes, but Chad, could you talk about angiosperms, gymnosperms, like mosses, ferns? Like, how are these? I mean, how are they, or if they're not, different in terms of their genomic architecture? What do we know about this sort of thing? Like, for example, I can say from metazoans, birds tend to have smaller genomes than mammals for some reason. There's some hypotheses, right?
Yeah. So I mean, you see kind of this variance as well. So like things like gymnosperms, think like a lot of your pine trees, that sort of thing. Angiosperms are all your flowering plants, so that's the majority of plant species. And then you have, I'm sure people are familiar with things like mosses and ferns, and like some of the genome level differences we see. Gymnosperms, to give an example, have huge genomes. We're sometimes talking some of the largest genomes out there. If the human genome's around, what is it? 3 billion base pairs, 3.5. I can't remember. I'm used to
very low threes now, I think.
Yeah.
Estimate.
Then like some of your gymnosperm genomes are more in the order of 40 billion or 60 billion, right? So 10 times or more the size of a human genome, but they tend to be mostly diploid, and most of this massive genome tends to be highly repetitive transposons. Your angiosperms show an incredible range of genome size, from some very small ones like Arabidopsis, that model species I mentioned earlier, 150 million base pairs. Something like corn is more human size, around 2.5 or so billion base pairs. Wheat is more in the order of 16 billion and on up. But angiosperm genomes, that's where you find a lot of the polyploidy in plants. Pretty much every angiosperm that exists has some history of polyploidy. Maybe it was millions and millions of years ago. Maybe it's very recent. And many of them have had multiple rounds of polyploidy. And then when you start talking about things like ferns, you can get crazy situations where they have huge numbers of chromosomes, not necessarily due to polyploidy, but just lots of small chromosomes. And then like things like some of your mosses tend to have smaller, kind of more simplified genomes, I'd say. So, but yeah, there's quite a bit of variation in kind of the genome architecture that exists across different plant species.
Well, I mean, you mentioned transposons, and as the host of this show, I need to elucidate facts for the listener. Not everyone knows what a transposon is. Can you tell us what a transposon is? And by the way, for the listeners, transposons are a big deal in the human genome too.
Yeah, so transposons are these selfish genetic elements that essentially make copies of themselves throughout the genome, kind of irrespective of the effect on the genome. So, if a transposon makes a copy of itself and places itself within a essential gene and kills the plant, well, that's the consequence, right? So they just make copies of themselves as much as they can until the organism tries to turns them off. So like plants and animals both have extensive, complicated machinery that exists almost entirely just to turn these things off and to prevent them from spreading throughout the genome. And they can take many forms and spread and copy themselves in many different mechanisms. New types of transposons continue to be discovered, but these were originally discovered back years ago by Barbara McClintock, one of the few plant biologists to win a Nobel Prize. She was doing this in maize, and the whole one thing you oftentimes hear transposons called is jumping genes, because the ones she would study would like basically cut themselves out of where they were in the genome and pick themselves up and move themselves to a different location in the genome. And so the effects of these can sometimes be observed visually just by, say, looking at the color of kernels in corn, where you see this variegation in the color of the kernel, and that's because in different cells that that transposon has picked itself and moved itself into a different location, and that affects the coloration. They've been extensively used for genetics, for genetic screens. They play a huge role in the evolution of species, both animals and plants. I think I'm a little iffy on some of my mammalian biology, but I think like placental development in mammals, it's due in part to transposons, right? And obviously,
I think I've read that
in plants as well.
I mean, okay, so I know you're a molecular geneticist. I mean, you do genomics, you do a lot of different things, like a lot of academics, but a lot of your background is as a molecular guy. And one question that I have is there like no portability to the molecular mechanisms or genes, genetic mechanisms in plants to say humans? I know, Drosophila people and mice people, obviously, their whole communities are geared around this idea that they could be a model for human. I mean, they get NIH funding and all that. I don't think plants usually get NIH funding. I don't know, but maybe there's some pharmacogenomic stuff. But I guess my question is, how different are the molecular mechanisms in plants compared to, mammals? When you look at a given gene, some of the genes have to be conserved back like pretty far, right?
Yeah. So, I mean, it varies quite a bit. And I mean, even some of those conserved genes start to look rather different. But it's when you consider just basic cellular functions, right? The core mechanisms are much the same, to where you can translate some of these functions back and forth, right? To give a few examples, like ubiquitination, right? This is a whole pathway that's involved in modifying proteins. You add this chemical mark called ubiquitin onto a protein. Usually that signals that protein is going to be degraded. So it goes through this whole degradation pathway. Well, that is highly conserved between both humans and plants and flies. And in both plants and animals, it plays a critical role in signaling and controlling transcription factors, etc. Basic metabolism that you see with the mitochondria, that's all conserved. The general concepts of signaling and transcription and translation, all highly conserved, right? But there are key differences, and that vary quite widely. It just depends on what you're looking at. Obviously, plants have a lot of biochemical pathways that just simply don't exist in mammals. They're great chemical factories. A lot of that revolves around the fact that plants are sitting there in their environment and being bombarded by bacteria and pests. And so a lot of the ways they defend themselves in response is chemically, by producing something that will kill an insect. Think things like nicotine, cocaine, etc. Those are all naturally produced by plants to kill insects. Humans don't do that. We can, if an insect is biting us, we can get up and leave or smash it. But on the other hand, humans have an adaptive immune system that we don't. Plants don't. Sorry, I may be thinking about plant genomes too much and personalized.
Yeah, no. So I do want to say really quickly for the listener out there, transcription and translation. These are these fundamental processes. I think a lot of you already know, but transcription is basically going from the DNA to the RNA. So you have like a string, and that string is pulled in, to like a to a molecular machinery that translates into protein, right? And so I just want to be explicit. These are basic unifying mechanisms that occur throughout life, I guess. Now there are differences, as obviously Chad knows, and many of you know, between these processes and say bacteria and say mammals or like more complex organisms. So I'm thinking about splicing and other things like that. So there's a core molecular machinery that we're all kind of exploring, and that's why Mendel's P experiments, which showed dominance recessive dynamics, I mean, that's how we figured that out.
Yep.
And it's in plants, right? So the laws of genetics, quote unquote, are the same fundamentally, even though they obviously have their own details, and this is why we have specialities in there are people who know how to do mice really well, etc., who have like all these clonal things going on. Talk about genetics and agriculture. Obviously, in some implicit way, we've been doing it since the beginning. But then you have scientific genetics over the last 120 years or so. How has that transformed things? And then you have genomics, and that's a whole different thing.
Yeah. So, I mean, like when we think about genetics and agriculture, I mean, really a lot of the basis of genetics as we understand it was, I'd say, developed within agriculture, right? If you go back to the rediscovery of Mendelian genetics at the beginning of the 20th century and for decades after that, where a lot of the development, whether it be quantitative genetics and kind of classical genetics, was really geared towards agriculture. Not always in plants, sometimes a lot of that was developed in livestock, right? I mean, some of the best quantitative geneticists in the world now are, work in livestock, cattle, and that sort of thing. But quite a bit of it was developed in plants as well. And so, most of what we think of as the role of genetics in agriculture, a lot of it's geared towards improving crops, right? So I mean, or improving livestock. So breeding for higher yield is always the big goal, but there's also a lot of aspect of selection on specific traits, whether it's for defense, improving defense against insects, against drought, abiotic stresses, things that aren't living, right? Or producing specific things, desirable characteristics. If you're working more on things that are directly consumed by people, texture, taste, that sort of thing, that's increasingly become a big thing. A lot of the method, obviously you can, for many reasons, especially ethical, right? There's you can do breeding in livestock and plants that obviously does not go on in humans. And so there's a lot, there's a lot of the initial discovery aspect, which is kind of where I'm at. But then there's also a big applied aspect where you're selecting for those traits within populations to generate a population that really gives desired output. So that's a huge thing. But it's more than that as well. For instance, it's thinking about, in the case of crops especially, it's thinking about what are some of the biotic factors that plants face. So, we're used to thinking of human medicine, bacterial infections, obviously things like COVID-19, right? Viral infections. Well, those occur in plants too. Plants get infected by fungi, they get infected by bacteria, they get infected by viruses. And so understanding the genetics of those organisms is critical to understanding how the plants respond, or building plants that are resistant to those, understanding the genetics of insects, especially resistance to some of the mechanisms that we select for or engineer plants to resist those insects. That's a big deal. Same with weeds. So like kind of understanding these natural populations of plants that we want to eliminate and how they're resistant. And that can lead to very fascinating phenomena. For instance, many people have probably heard of Roundup, right? One of the most widely used herbicides, one of the most successful. And so there are a number of weeds over the years who have started to develop resistance to that. But some of the underlying mechanisms of this resistance is quite fascinating, even from a basic biology standpoint. So to give a little background, Roundup, scientific name glyphosate, targets a specific enzyme that's involved in amino acid biosynthesis. It's not that that enzyme is not found in any animals. It's specific. It's only found in plants and bacteria. One reason why it was such a successful herbicide. And glyphosate inhibits this specific enzyme. Well, one of the mechanisms that has evolved to develop resistance is by just cranking up the amount of protein that you produce of that enzyme to the point where you overcome the inhibition by that herbicide. One way you increase the number of protein is by increasing the copy number of the gene that encodes that enzyme. And in the weed Palmer amaranth, the way it's gone about doing that is it's actually formed these extra chromosomal or circular DNAs that have just pretty much that gene on it. And so these are circular pieces of DNA floating around in the nucleus that are not part of any chromosome. And these ecDNAs can increase to something like 50 or more copies within the nucleus. So you've basically increased your copy number like 50x of this gene, allowing you to crank out more proteins. So these ecDNAs have been found extensively within cancer. They play a big role there. So you have these very interesting kind of odd mechanisms that it can occur in an agriculturally important way that are not just the traditional thinking about breeding a crop.
Yeah.
Right?
Well, I mean, one thing that I'll say is, we talked about GMOs before. We're not going to talk too much about GMOs, but I will say genetically modified organisms, just for the people out there, people are freaked out about genetically modifying humans, obviously. They're also kind of freaked out about genetically modifying animals. But with plants, I feel like the threshold is lower. I feel like a lot of the techniques, plants are obviously different in some fundamental ways, but still, I don't know what you think, but I think a lot of the techniques are probably going to be perfected in plants just because the quantity of the reps of the, you can make errors in plant lines that, and then also with animals, I think the issue is people are freaked out. Like I've told this story before. When I was back at Davis, I had a friend, he worked with goats and fertility, and they were doing weird things with their testicles, and, they was on lockdown because they were worried about animal rights activists. They were worried about religious fanatics. They didn't know. Because like mutated goats, that's a tabloid headline.
Yeah, I mean, obviously a lot of the genetic modification has over years, right? I mean, like this is most of my life now. I remember when the first genetically engineered corn and beans started being rolled out, right? I mean, this is obviously garnered a lot of headlines, a lot of controversy or opposition over the years, but definitely, I mean, obviously, we've had transgenic, where you've taken a gene externally and put it in corn and soybeans for at this point around 30 years, whereas transgenic animals just haven't almost never made it to market. There was like a transgenic salmon that a company was trying for years and years, and I can't remember the state of that, but I think it's been going on for probably 20, 30 years, and they still really haven't done much with it, right? So, I mean, you're right, it's a lot more difficult to do, and we're obviously by now we've brought out a lot of transgenic crops. Usually specifically in those things like corn, soybeans that are used more for animal feed or industrial uses rather than direct human consumption, because then you've run into really solid opposition. Obviously, I think they're safe. But the beyond kind of the transgenic end, the genome editing, that's going to be rolling out, and I mean that's already starting to roll out in plants. You're starting to see it to some extent, I think, in livestock as well, but again, there's been a lot more barriers than what you've seen with plants, and it's going to roll. I mean, I think countries that used to be very difficult to deal with on the transgenic end, like places in Europe, are signaling that they tend to be more open on the genome editing front. And so I think this is going to be rolling out big in, I think, a lot of crop species, and probably even in some of the foods we consume directly.
Wait, why are they more open on genome editing than transgenic? So, I mean, just to be clear, transgenics is you take a gene from, one lineage, you put it in another, Whereas genome editing is you go in there and, with CRISPR, like you could get, in theory, really fine grade base pair changes, right?
I think it's kind of the side. I think it's like going from moving an entire gene from one organism to another versus going in and making a very targeted modification. I mean, the reality is the former is actually easier than the latter until very recently. But I think the way people see, like, oh, you're making just one small little targeted change versus moving this whole thing, like, I think it's just a perception of one being more drastic than the other, even though that may not be in reality. When you think of the effect on the plant, that may not be true at all.
Yeah, and just to be clear, the average human gene is tens of thousands of base pairs, right?
Yeah.
There are genes that span millions of base pairs, although a lot of that is probably intronic. But in any case, genes can be big, and as opposed to doing a modification on a site, I can imagine that sort of thing. Some like a regulatory element, like you modify a base pair, and that is that affects the regulation in some deep way. And there's like a huge phenotypic effect. How has so we're talking about genetic engineering, which is writing the genome. How has having access to the whole genome transformed agricultural genetics?
I think it's been huge. So I mean, like, if you think of kind of basic things that have been done forever, like breeding and identifying quantitative trait loci, right, to select on in breeding, you've gone from kind of these old school kind of painful to put together genetic maps, where it's like doing this took identifying a single QTL, a single trait that you might want to breed for, could be an entire PhD. It could be years of work, right? To where we have the entire genome, we have huge density of variation across that genome from things like whole genome sequencing that just never existed before. And that allows you to then map those traits much more quickly through methods like GWAS, etc. But beyond that, also kind of where it's moved in the breeding end is that's gone from kind of this older approach of kind of selecting first on phenotypes from multiple crosses to marker assisted breeding, where you had a marker associated with a specific trait and you selected on that, to what's called genomic prediction, where basically you're taking all the variation in the genome and you're calculating a score that is predictive of the trait of interest based on a bunch of crosses purely based on the sets of variants in that genome. And then that allows you to select the individuals that you want to continue on. And that is so much faster, and it's really sped things up quite a bit. And that's just on the breeding end, let alone on the molecular, like kind of more the biotech end, where usually you're trying to identify specific traits to, like identifying those specific loci. Maybe you want to target with genome editing for a specific modification. So it's really sped things up, where I know certain things that used to take, a decade might now take a couple years, right?
Yeah, I mean, so you're talking manifold increases in productivity in theory.
Yeah.
And so you're erstwhile academic, you were a professor at a Research One university, and now you're in industry, and not your goals are a little bit different now, a little bit more applied. Obviously, they gotta hit towards the bottom line. So are you feeling good about the transition? Like, what's the biggest difference? Like, just give me a sense.
I mean, I think the biggest difference might be a little bit obvious. Now I used to be in academia. I was kind of split many ways. I ran a lab. I was doing research, funding that research. I was teaching, and that the teaching becomes a huge effort, right? And so I mean, like, when those semesters when I taught, my research efforts sometimes would just kind of be like maintenance mode, where I'm just kind of making sure the lab is still running, but I'm not really pushing things very hard until I'm no longer teaching. In addition, I would be doing things like service, serving on committees, some of these very tangential or unrelated to research, or even sometimes teaching. You had this expectation of being involved with societies and going and giving talks, etc. And then increasingly other changes like what did you do for DEI and all that sort of thing. Whereas now I'm almost entirely focused on research. It's not to say that I'm not busy, but I am 100% focused on research, which is really kind of what I've learned I love doing. And so that change has been has made my quality of life a lot better, right? So the pay is better in general as well, so that always improves the quality of life as well. And so I think I've achieved a better balance between doing the work I really am passionate about and also being able to spend more time with my family, which was kind of the big kind of deciding factor for me. Yeah, I mean, there's a lot of other aspects there, but the change has been wonderful. So I know some people probably regret that, and I know some people have gone back into academia.
Well, I want to ask you a little bit more of that, but one question that I do have, I know in artificial intelligence, for example, but basically a lot of computer science, public-private collaborations are just basically a necessity because, places like Google have resources which no academic lab or academic cluster, like even Stanford, cannot match. And so I was just curious, what is the scene like when I was at Davis, and I think you did a postdoc there at Davis too, public-private partnerships were a big deal. And that's just because, there's a lot of plant geneticists there. And yes, it's a basic science, but we grow plants to eat them. There's a huge agricultural sector. What is the state of academic private sector collaboration in your field right now?
I think it varies a lot. There are certainly I think on the academic end, it varies considerably based on kind of really the nature of the department you're in. So I think you'll find that if somebody who's kind of more in like the basic departments, like say a microbiology department or just a plain plant biology department, you might see a lot less of this sort of collaboration. And I think a lot of that is simply due to the fact that a lot of the individuals in those departments are not even aware of the opportunities for collaboration. Whereas if you go to departments that have a lot more of kind of the applied end, like horticulture or agronomy, those sorts of collaborations are quite extensive. And sometimes those collaborations are a necessity to the faculty or the researchers in those departments, where maybe the resources, the funding available for the sort of very applied research that they do is just almost non-existent in the public sphere. They can't, if they go to something like they can't go to the NIH, they can't go to the NSF. The NSF says this is too applied, and the USDA has limited funds. So the companies are their lifeline sometimes. And yeah, so it varies considerably. And then it just kind of depends, right? Obviously, the sort of what a company is willing to fund is going to be, is this something that is relevant to what we're doing? So they're not going to be funding any basic research. But if they are, it's something that's going to be quite critical to their kind of plans. But they also fund a lot of applied end research that simply wouldn't happen otherwise. Things like a lot of weed scientists, like studying them out there, studying weeds in the fields, right? That's something that, a lot of industry is willing to fund, but very little of the public sector is.
Yeah, that makes sense. So going back to academia, you've been out of it for a little while. So now you're an outsider observing, and you were an insider for a while, and you had a lot of opinions. And now, I know you're in touch with people in academia, and I'm sure you still have few projects that you're probably tying the bow on and whatnot. How are you feeling now? Look, you're happy. You're very clear about that. You're happy where you are. But just in terms of more granular level, how are you feeling about where academia is now versus where it was when you were there? Just candidly, I know that you have talked about, like, over your academic career, there was a, let's say, maybe a decline in the skill level of the undergraduates on just basic things like math. And I think I'm comfortable saying this because pretty much every other professor I've talked to has said the same thing. So I don't think it's a delusion.
Yeah, I mean that's certainly the case. My feeling, I mean, I don't think you're going to see much change in academia anytime soon, especially as long as there's huge sources of money from the public, the NIH, NSF, etc. Things aren't going to change in academia anytime soon. But I would say that, over the years, I've kind of soured on a lot of just the academic model in general and what drives a lot of academia. I mean, there's the amount of bad research, honestly, that is published on a regular basis and is quite astounding, right? And that's driven by kind of these perverse incentives that academia has really set up for things like tenure and promotion, and that the funding situation has set up where you had, I mean, lots of people have talked this, right? Like publish or you perish sort of deal, right? And it's there's definitely great research being done. There will continue to be great research being done, but there's a lot of bad research that is done as well. There's a lot, and I think that's going to be more challenging as we continue to, like, the quality of a lot of students, I think, is going down, or at least when they're coming out, they're not as knowledgeable as they should be, and that's going to play a role also in that. It's academia needs some major reform, in my opinion. It can be a great place, it can be a wonderful environment, but it seems like it's decline that has declined considerably, but I don't know, short of a major shock, how that's going to reverse course anytime soon.
Yeah, let me just for the listener out there, I'm gonna surface just some conversations I've had with people inside and outside of academia. Some of them, Chad, but and these are public data, but people don't think about this too much. The number of students is dropping, and that's just because people are not having children. So this is a structural fact. In the 2010s, the teens, this was compensated in part by Chinese students, I think, quite clearly. Yeah. Those students are not coming anymore at the same quantities and levels, partly because the Chinese themselves are declining in numbers. Also, partly because there's some better opportunities in China and the geopolitical tensions that are making it more difficult. They'll still come, but it's not the cash cow that it was. So you have a declining number of students. Like maybe they'll get students from India, like Australia and Canada do, where basically a lot of their student visas are fake. Like, just candidly, I'm going to say it. Maybe that's what universities are going to have to do. And, a lot of academic scientists don't realize, but, tuition is really necessary for these universities because you have all these fixed inputs, but they're all accounted for. So when you remove money on the margin, how are things going to get paid for? So there's going to have to be cuts. A lot of these universities are over leveraged, so they're in debt because they built these facilities to attract students. And so I think what I don't want to use the word neoliberal, but I'm going to use it. It's a model of education as consumption. And the problem here is, look, when I was an undergrad, I didn't like doing homework either. I would have liked, I'm not gonna repay the full freight on the rock climbing, whatever. So there's all these things that are being created as extras, and that's student driven. And also now we're having a situation where they're going to compete for a smaller and smaller number of students. What's that going to do?
So I can tell you what it's going to do. I mean, and this is something that has made me very angry, kind of very soured me on academia over the years, is, here's an organization that's supposedly like this nonprofit, right? But it's all the internal incentives are very profit driven, right? And so it's almost like become profit already and just be honest about what you really are. But to give an example, right? I mean, like, what's going to happen at least is the larger universities are going to prey upon the smaller universities and drive them out of business. And that's going to keep them afloat for a while. And it's going to drive, like, you already hear about these small liberal art colleges all going out of business, right? Or, going bankrupt and closing because they just don't have enough students. I mean, when I was Michigan State, I'll be frank, like one day the dean came and talked to us, and he basically told us, the university has a plan to basically double the enrollment. The way we're going to double that enrollment is by lowering the admission standards and taking students who would normally go to kind of the smaller schools where they would have smaller classes, more personal, and they're basically going to poach those students. And one of the things he told us is that, well, you're going to have to teach students who maybe are less prepared in terms of their writing ability and their math ability. I'm like, okay. What additional resources are we going to get? This was at a time when we were getting regular budget cuts, right? So it's the internal dynamics of academia is being driven very much by business considerations, which, when you think of a nonprofit university, that's not what any of us want. But tuition's going to go up, and the larger universities are going to poach from the smaller ones. And you're going to see more universities go out of business. And what the longer term plan is, when that well dries up, I don't know.
Do you know? There's some science fiction. It's some British thing. But they have something called Municipal Darwinism, and it's like these like cities, mortal engines. Okay, I didn't read it, but there was a movie about it, and it's like these cities swallow up smaller cities and assimilate them. And what you're describing looks like it's going to be academic Darwinism, where they're going to just cannibalize all the students from Western Michigan or wherever, and probably they're going to have to, there are smaller state schools that have shut down as well. It's mostly private schools. Yeah. My understanding is the ones that are really on the chopping block are the schools that have like high tuition, low name recognition, but they're schools that people have traditionally like gone to their families or something. They have some connection, but people are making an economic calculation, and they're like, they're going to go to the flagship because it's cheaper, it has a bigger name recognition, or if they're going to spend the money, they're going to go to Harvard or maybe Wesleyan, or that's like on the bubble in my opinion. But there's a lot of small, good private liberal arts colleges out there that no one knows about.
I mean, everybody knows that there's a hierarchy in academia, right? I mean, like everybody knows Harvard, Yale, Princeton, those sorts of places. And then like a lot of your top tier state schools, and then even on down. And then when you start getting into your R2s and below, right? I mean, those are oftentimes not recognized. And there's a lot of academic snobbery, both within and outside of academia, about what school you went to, right? I mean, like job searches where, people coming in like they went to
we call it pedigree, right?
Yep, exactly, pedigree. And so that's going to play a role in the decisions that make as there's fewer students, and those places try to maintain enrollment or increase it to affect their bottom line, and that means those at the bottom of the are just going to disappear.
Yeah. And also, let's talk about like what's happening to academics themselves. The demands for publishing have like been ratcheted up a lot, and honestly, this isn't necessarily greater like real scientific productivity. It's just you need a line on your CV. Yeah, so just like push stuff out, And you said research too. So research 2 universities traditionally were teaching universities more. They were like the second tier. They're not research 1. They're not like, and like the Pareto principle applies in academia. Like 20% of the institutions produce 80% of the output. Yeah. You could remove the bottom 80% and still not be that different, right? Well, so these research 2 universities, now they're demanding their professors do some serious research, which is fine, but like, it's kind of fake.
Yeah.
Right? I mean, in terms of like,
And I mean, the thing is, they're demanding that their professors do more. And unfortunately for those faculty, right, they usually have very high teaching loads, right? I mean, they're teaching every semester, sometimes multiple classes per semester. The expectation that somebody can, I would teach a large undergrad course of, 180 some students, right? That takes a lot of time. And that'd be one semester, and then I have a semester off to do research. But my research, like I said earlier, would basically just grind down to maintenance mode, where I'm personally not contributing a lot to just kind of making sure things are keeping rolling, while most of my time is consumed with teaching. And the expectation that those faculty, when they're teaching sometimes multiple classes each semester and sometimes during the summer, are going to have a serious research program it's unrealistic and it's unfair to them. But even at the higher level and stuff, like there's additional demands always being put on. Like there's always the service component, right? Why aren't you serving enough on different committees? And those may not be a huge time, but it adds up and contributes to that. And then throw in, the last year and a half or 2 years that I was at Michigan State, suddenly the tenure requirements change. Now, in addition to service and teaching and research, you also have to be contributing to DEI. Like, okay, what does that even mean? What does that look like? I won't even go into kind of like the my disagreements there, but it is additional workload being added on to jump through more hoops to achieve this ideal goal of being the tenured faculty, when you have all the freedom in the world, right? A secure job. And I'm like, it's
so what is first of all, I'm going to say, like, because I know this because you told me, you guys were not getting paid more.
Not really.
You guys were being asked to do more every single year, and this is at a time of like the beginning of inflation, and like you get budget cuts,
Yeah. I mean, you would get pay raises, but I mean, moving to the agricultural industry, my pay increased by almost 33%, right? And all I have to do now is just do research. I mean, you got pay raises, but they weren't huge pay raises, but the amount of work you're expected to do increases, and a lot of people succeed, and they put their head down and they get it done. And I just kind of say, I have other priorities.
Yeah, I guess I guess the last question I want to like talk to you about is I feel like over the last 15 years, academia has changed a lot. I was involved from 2011 to 2016. I still have a lot of friends in academia. But I feel like even over those 5 years, it started changing. I left before the really big changes in the late teens and after 2020 with DEI. Very, very political. I mean, did you perceive that as well?
Yeah, I think it's definitely politicized. I mean, I don't want to go into too many details, but I think it's become a more politicized environment. Maybe political isn't the right word, but definitely a more politicized environment. Definitely a more conformist environment. I would have been an undergrad starting back in 2003, right? And I remember as an undergrad in a grad school, having full-on arguing with my professors, and that was tolerated in a way that I don't think would be now, right? And I saw a lot of professors during those years very openly express political opinions that I disagreed with, but it was just kind of accepted that this is a place where free speech reigns, right? And I think that's definitely not the case anymore. It's definitely become more politicized and more miserable, I would say. I mean, maybe it was just my perception, maybe because I was miserable towards the end, just getting miserable in academia, but I didn't really think that my colleagues were happier either, even though I know that they would be on the opposite political spectrum. Like, they didn't seem happier either. But maybe that's because we were faculty then and we're all stressed out.
Yeah, it's kind of sad because this whole dream of like tenure and all that stuff, but the reality is you're in a institution where there's other people, and one of the upsides for a lot of people is the collegiality of like being among peers. If you're a scientist, you have to collaborate.
Yeah.
at this point. For the vast majority, you could be like really abstract mathematician and maybe you do your own thing. There's a few people like that. But in general, natural scientists have to collaborate. You need collaborators. And so when you're talking about conformity, I think part of the issue is like, it doesn't matter if you have tenure. It doesn't matter if you're a full professor. To some extent, it doesn't even matter if you're like fully funded for like 20 years. You have to place your graduate students. You have to help your postdocs get jobs. You have to be on good terms with your colleagues. And so you can't be totally independent, or if it's not tolerated. So I think what I would say is my perception, because I don't have like, I wasn't, there. Well, I mean, I was an undergrad in the 90s. I'm a little older than you. But my perception is there was a culture in academia for many decades that oddballs and ornery people, that was part of the game. Now, in theory, they could have ostracized those people, but they didn't because, well, this is just the place where the weird people are, and you have to accept them no matter how weird they are. So I'll give you a concrete example, and I know that you're not a big fan, but like Peter Singer, the utilitarian philosopher, he's a big deal. He's highly cited. He's also defended infanticide.
Yeah,
like very few of his colleagues agree with that, but it was kind of accepted. Like, okay, he's weird and that's freaky, but we're academics, there's communists on the faculty too, whereas like now over the last couple of years, there have been like some serious protests about Singer speaking at campuses because, he's against disabled people, whatever. And so I think that's what I'm saying. Like Singer is kind of like the outermost, like, and he's very autistic. Well, I don't know if he's literally autistic, but from people who have talked to him, he is just not appropriate. In terms of like the things he says. I mean, normal people, even if you think about that, you're not going to say it, but he does. And so it's in keeping. He was ensconced and had a relatively high status in academia for decades. And now they're trying to cancel him, you know? And I think that's the change. Now, he's the tip of the iceberg. There's many people, much less controversial, much more small potatoes, but I think the way they react is just keep their heads down and don't say anything, know?
And I think it kills a lot of joy. I've always, when I was a faculty member, I believe very much in kind of, being professional, right? It's like I'm not one that thinks that politicized discussions have much of a role in the lab or the classroom unless there's a real reason for them. And as a plant biologist, usually there's not really a real need for them. So I'm usually, you keep your opinions to yourself, I keep mine to mine, and we'll be happy. And that's mainly because I like avoiding controversy. But there is kind of a misery that comes from just knowing that, like, even if you wanted to, there's going to be consequences if you speak. And I think there's less freedom. There's less intellectual freedom than there used to be. And it's unfortunate because I think that's essential to a really successful and high functioning university. But
yeah, I mean, we can't change it on our own, but we have kids, and like we'll see how it is in a couple of decades. And people vote, people engage in activism, people donate money, and so ultimately it's up to us. It's up to us in the aggregate. We can only do so much, but if everybody acts, if enough people who have concerns act, there will be changes. And things that are not sustainable will not sustain at some point. Now, it could go on for a while, obviously. To use an extreme example, Soviet communism went on for 70 some years. It already had problems in the 1920s. But it went on and on. Arguably, OPEC, the oil shocks of the 70s extended it. But, that just shows you things can go on for a long time. But, they end. And so I guess like that's where I want to end this conversation. There is hope, that there can be a correction. And, the stuff we were talking about earlier, we're talking about plant genetics, we're talking about transcription, translation. Those things are real. Those things are out there. Like reality and truth do not change. Humans change. Our cultures change. But the world out there that we explore with science and scholarship do not change. So I think that's what I would want to end on a hopeful note about, that like nature is out there and we need to seek it. And if we choose not to, that's on us. That's on us. But we have the ability to do so if we choose to do so. So let's create a culture of truth again, at least in your local space. Like everyone needs to do it, even individually. Like start small. Speak up when you can. Don't be insane. Don't be out in the street so you can't support your family. But like little courages together can cause a lot of courage, in my opinion. And the people that you're talking about, Chad, who kind of keep their head down, you're alluding to the conformists. The reality is they're not strong people. So if enough people with courage speak up, they will join us because they just joined. So that's my hopeful note, bro. All right. So it was great having you on. I will see you around. I'll see you around online, and good luck on everything, man.
Yep, you too. Bye.
Even if you and your partner are healthy, there's still a chance your child can develop a serious genetic disease. This is because every embryo has new changes not present in either parent. Most of the time, these are benign, but sometimes they can be catastrophic. Orchid's whole genome embryo reports directly screen the embryo and analyze these de novo genetic mutations. Discuss embryo screening and IVF with a genetics expert.
This podcast for kids. This